Everyone. So today we're going to be continuing with our series on biochemistry. We're going to be talking about amino acids and proteins. So roughly 15% of the body is combined of proteins. Is crucial in almost every aspect in every part of the body. So biological, metabolic pathways, so synthesis of other molecules, uh, uh, catalyzing other reactions, and also proteins form the structural components for a lot of different cells or a lot of different body parts and organs and stuff like the protein that makes up the the gel structure that's in your eye, for example. And so proteins, usually one thing we see with proteins is that function and structure go hand in hand, right? And so one example of this that we can look at is we can have uh, what are called prion proteins, and these are basically proteins that are misfolded. So in this normal protein here, we have three different elements of secondary structure. These are all called alpha helixes. Then this is a cartoon depiction, but it's very common in biochemistry to see proteins drawn this way. Three different alpha helixes. And then in the diseased protein, we see the only thing that's changed is that the alpha helix, one of them is not there anymore, and now we have what's called this beta barrel type structure here, right? And so this is the exact same amino acid sequence, meaning that each amino acid is identical. It's just that the way that it's folded in three-dimensional space is different, right? And so this is called a prion protein, a misfolded protein, and it can induce other proteins to misfold. And so this can lead to a series of neurological disorders. So, for example, mad cow disease, uh, zombie deer disease, which you guys may have seen in the news more recently. I'm spelling that wrong. Uh, these different diseases are caused by misfolded proteins that then can induce other misfoldings in other proteins, right? So proteins are everywhere in biology, and in my opinion, the most interesting and one of the most important biomolecules that we look at. And so there's a hierarchy of structure in proteins. And so the function of a protein is determined by its structure. So again, we're seeing that structure and function correlation. And proteins are built on what are called amino acids. And every single amino acid has the same basic structure except for one part that we'll talk about it. And so the structure for the hierarchy is we're going to have primary, secondary, tertiary, and quaternary structure. This hierarchy, so how do we determine the different structures? Is basically we're going a step out and looking at as the structure gets more and more complicated. So primary structure is just the specific amino acid sequence that we see in proteins. So the same way for RNA and DNA, we might have a sequence of AAGC, for example. In protein structure, you might have a sequence of arginine, lysine, glycine, glycine, something like that, right? And so this is primarily held together by covalent bonds. So these different amino acids are snapped together by what's called a peptide bond, which is an amide functional group. And those are covalent interactions between those. So this is probably the strongest level of protein structure, the hardest to get rid of. We have to have chemical reactions to break primary structure. Secondary structure is going to be interactions between different amino acids within a peptide. And these are often going to be that are not neighbors, meaning that they're not neighboring amino acids. They're going to be separated by other amino acids in a single protein. And these are generally held together by hydrogen bonding, specifically hydrogen bonding of the protein backbone of protein backbone. And it's also held together by disulfide linkages, which we'll talk a little bit about. This is basically for cysteine. You can have two cysteine molecules get reduced to form a linkage between two cysteine amino acids. And so this is structure that's held together by uh, hydrogen bonding and disulfide linkages. And two examples of secondary structure that I want you guys to remember is going to be the alpha helix and the beta sheet. And so the alpha helix is what we saw in the cartoon. Looks like a helix like that. This is going to be an alpha helix. Then a beta sheet is going to look like something like this in a cartoon structure. They're usually signified by arrows, and they go back and forth like this. And the hydrogen bonds are going across this way in a beta sheet, and an alpha helix. The hydrogen bonds are going to be along the helix like this. So this was a beta sheet. So those are two elements of secondary structure I want you guys to know. Tertiary structure is going to be interactions between secondary structure within, and this is the important part, within a single polypeptide. Right, so if they are part of the same amino acid sequence, then and it's an alpha helix interacting with a beta sheet, for example, that is going to be tertiary structure. And this is primarily held together by hydrophobic interactions, charge interactions, and you can have other things like hydrogen bonding, but it's primarily held together by these interactions. Quaternary structure is going to be interactions or forces between different polypeptide chains. So we can have the same type of interaction between two alpha helixes. It can be tertiary or quaternary structure, but quaternary structure means that it has to be between two different chains. So if we have something that looks like this would be an example of tertiary structure, and these would be the interactions between the two alpha helixes there. And this could be an example of quaternary structure because each color here represents a different chain. This would be quaternary structure. And it's just because we have two different chains, even though the nature of these interactions are exactly the same. All right, so those are the different structure classifications for proteins. We have primary, secondary, tertiary, and quaternary. You can think of it as you start to pull a protein apart together. First, you're going to pull apart the quaternary structure, then you'll pull apart the tertiary structure, then you'll start to unravel the beta sheets and the alpha helixes, and eventually you'll start to pull on the covalent bonds, which is primary structure. That's how I like to remember the hierarchy there. And so let's look at elements of primary structure then. First, so primary structure is the specific amino acid sequence. So we should talk about what an amino acid looks like. It has an N terminus, and I usually think of nitrogen as green for whatever reason, and a C terminus. And these are going to signify the ends of both a polypeptide and a single amino acid. And so usually at neutral pH, the N terminus is going to be protonated like this. This is connected to what's called the alpha carbon, and this is going to be the stereocenter for the amino acid. And then that's connected to the C terminus, which is usually deprotonated at neutral pH. And on the alpha carbon, we have the R group, right? And so the R group is important because the R group is called is basically what gives the amino acid its identity and all of the properties of the amino acid. And so this is connected to the alpha carbon. And this R group, again, this gives us the identity, meaning the specific amino acid that we're talking about, and the properties of the amino acid. And so the amino acid is generally broken into three parts again. And when we look at polypeptides as well, this will follow the same trend. We have our N terminus, our alpha carbon, our C terminus. The alpha carbon is our chiral center and is connected to the R group, which is given the amino acid its identity and all of its properties. And so everything that I'm circling in blue here, this is the same for every amino acid, right? So this is the same structural characteristic of every single amino acid. The naming of amino acids, again, is based off of the R group. Naming, it doesn't follow any particular IUPAC naming. These are all given by common names that are based on the R group. So for example, if we have an amino acid that looks like this, I'll give you, this is going to be H2 alpha carbon H and the C terminal of the amino acid. This R group is proline, and it is actually bonded to the amine side as well. And so the name for this is called proline. It has a three-letter sequence identifier that is PRO and a one-letter identifier that is P. We also have another example here. We can look at glycine. So glycine is special because this is the only achiral amino acid, meaning it does not have a stereocenter. And so glycine looks something like this. H3N, we're going to assume neutral pH right now, so it'll be protonated. We'll talk more about this in a little bit as well, but this will follow the same trend as when we talked about amines and how they could be protonated or deprotonated, and carboxylic acids, which will keep this one deprotonated. Glycine, the R group for glycine is just a hydrogen. So this is the R group. This is the R group for proline. And the three-letter sequence for glycine is GLY. The one-letter sequence is G, right? So this is the one-letter identifier, and this is the three-letter identifier. And then we have the full name, which is going to be glycine. So this is how we name different amino acids. Again, they're all common names that don't follow any specific IUPAC naming convention that we've seen before. And so how do we classify amino acids? Well, basically they're classified on their physical properties, which is determined by the R group, like we've already stated. And we generally break them into three different categories. We'll have polar charged amino acids, we'll have polar uncharged amino acids, and then we'll have nonpolar amino acids. And so again, this is all related to the R group. So is the R group polar? So we say, is the R group underline question mark, right? So is it polar charged, polar uncharged, or nonpolar amino acid R group? Sorry, no amino here. And also so basically a polar charged R group, an example of that would be a pH 11 solution. The R group can be, if we have the alpha carbon here, this will be, oh, minus that would be a polar R group. This would be an example for tyrosine. This is what the R group looks like for that. This goes off in other directions. A polar uncharged amino group would be like serine, where we have the alpha carbon. Then we have an OH group bonded. Sorry, I added extra carbon here. So this is the alpha carbon. Then the OH is directly bonded to it. That would be polar because of the OH group. And then a nonpolar R group would be something like, so this was serine, would be something like valine. And valine is going to look like an isopropyl that's attached to the R group. I'll write the name somewhere else. That goes to the rest of the amino acid or the protein backbone. and this was valine, right? So we classify the different R groups based off of their physical properties. And so how would we classify these three R groups here? Well, we see that this R group and the first one, we have an OH group, and the rest are carbons, but it's short chain carbon, right? Anything less than 5 carbons is still probably going to be pretty polar. So this would be a polar uncharged R group. Arginine, if you look at it here, in neutral pH, I know because I've worked with this one a lot, it's a long R group, but it has a positive charge on the imine group right there. So this is going to be a polar charged R group. And then we have leucine, which looks like valine, except we have one extra carbon. It basically looks like an isobutyl group on the amino acid. This will be a nonpolar R group. And I didn't write it there, but the nonpolar R groups you'll sometimes see separated into two groups. You'll have aliphatic, which are just regular sp3 carbon chains, and then you'll have aromatic, where you'll have the aromatic rings involved. And there's a series that we've talked about different heterocyclic rings that can be aromatic, benzene rings that can be aromatic. So those are all under the aromatic R group. We don't, it's not that important, but sometimes you will see that these different classifications within the nonpolar R groups. Right, so that's how we would classify these different amino acids. Amino acids also have stereochemistry except for glycine, which I mentioned before. And so, similar to carbohydrates, we have D and L isomers, and specifically they are enantiomers because they form chiral mirror images of each other. Again, these were called enantiomers. I don't know why I wrote it twice. The distinction between these, instead of the carbohydrates where you saw it was the position of the hydroxyl group on the penultimate carbon, these are going to be based off of the amino group in an amino acid. And so opposite to carbohydrates as well, where almost all naturally occurring carbohydrates were in the D configuration, almost all amino acids are going to be in the L configuration. All natural amino acids are going to be in the L configuration. And so if we look at an example, we can draw a Fisher-like projection for a amino acid. And the way we draw the Fisher projection is the same way. We put the orientation of the carboxyl and carboxylate group going up top. I erased that just because I want to stay with the same color convention, so that we O minus there. That then goes down to what our alpha carbon is. I'm going to give it a little more space because arginine is a very long R group. And then below we have the R group, which was CH2, 3, and then that was bonded to an N, which is then bonded to another carbon that was positively charged, and then NH2 here. This is what the arginine R group looked like. And then the amine, or the N terminal, goes off to the left here, right? So we know that because it's L-arginine. So that's how we draw the Fisher projection for an amino acid, right? So this is going to be the same as it was for the carbohydrates. So this was the Fisher projection. So these are all going to be L configurations for amino acids. And we didn't get to it this semester in the lab, but we can also do an optical rotary experiment where we generate a racemic mixture of both of the different enantiomers for amino acids when we do the benzoyl amino acids lab. And then we could generate the pure L-arginine. We can look how they rotate light differently based off the mixture. And that's a pretty interesting experiment to do based on polarimetry. And so we can do this for all these different amino acids. So this is just good practice so that we can become familiar with our amino acids. So we have all of them are going to have a C terminal. And then we have that go down to the alpha carbon. I'll explicitly draw the alpha carbon here because I want to. And then we can't draw the hydrogen yet, actually, because all of these, one of these is going to be a D isomer. So the two L's, we're going to have the hydrogen on the right because the amino group is on the left. We have H3 N plus. We'll just assume that it's a neutral pH. H3N plus. The D-phenylalanine, that's going to be on the right-hand side. And then we have the different I groups, R groups that go down. Leucine, again, was 2 carbons, an isobutyl group that's going down. Methionine is just a methyl group, so I'll just put a CH3 there. That goes down. And then D-phenylalanine is going to be an R group that has a aromatic ring, so this would comprise one of the aromatic nonpolar groups, something like that. And these are all drawn in the appropriate configuration according to the enantiomer that's specified there, right? And so again, Fischer projection, we're looking at the structure from the top and then smooshing them down. Okay, and so similar to when we talked or had our discussion on amines, where they can act as bases, amino acids, as the name suggests, can act as both acids and bases. And so the protonation state of the amino acid depends on the pH of the solution. The standard pH protonation state is going to be at neutral or physiological pH, which is around 7.1. At high pH, amines are, well, amines and carboxylic acids are deprotonated. Deprotonated, which leaves a negative charge on carboxylic acids and a neutral charge on amines. And at low pH, amines and carboxylic acids are protonated, which means we have a positive charge on the amines and a neutral charge on carboxylic acids. And at neutral pH, this all depends on the pKa of the different groups that can be protonated or deprotonated. So if we remember, the pKa is equal to minus the logarithm of the acid constant, which is equal to minus the logarithm of the ratio of the H3O plus concentration times the deprotonated form of whatever our acid is over the concentration of our um, protonated acid. And rearranging this and doing some algebra, we could see that the pKa minus the logarithm of the ratio of the protonated versus the deprotonated form is equal to the pH of the solution. So the ratio of these, if we then rearrange this a little bit more, so we did minus the logarithm, we could see that the ratio of the protonated to the deprotonated form is equal to the pKa minus the pH, all under the 10th power, right? So this ratio is affected by the pH of the solution because the pKa's are fixed values for the different amino acids. And so what that means is even though we have these general trends at high and low pH, we can actually look at what the protonation state would most likely be depending on the ratio of the we can just looking at the pKa values for the different groups. And so what I'll be giving you guys is something that looks like this, where you have pKa values associated with all the potential protonated or deprotonated species on the amino acid. And then from that, you can look at the pH of the solution that the amino acid is in, and then determine what the protonation state would be. So if the pH is less than the pKa, then the protonated form is going to be favored. And if the pH is greater than the pKa, then the deprotonated form is going to be favored. And so I recommend following a little mental flowchart when you do this. So what the mental flowchart will look like is the first step is you are going to be looking at whether the pH and the pKa are how they compare to each other. So if the pH is greater than the pKa, we said that we favor the deprotonated form. And if the pH is less than the pKa, we favor the protonated form. Then the second step of the sequence is we have a chart here that'll say protonated and deprotonated. And we have our different elements. We have our amines and our oxygens that'll be protonated or deprotonated. When we have a protonated amine, we have something like NH4, has 4 bonds, so it has a positive charge. These bonds can be taken up by alkyl groups as well, or R groups. This is going to give us a positive charge. And the deprotonated form is going to be something like NH3, so this will be neutral. The oxygen we will have in the protonated form HO, which is neutral. And then the other bond is taken by whatever else it's bonded to, most likely it's carboxylic acid. In the case of tyrosine, it would be the benzene ring that is bonded to. And then in the deprotonated form, we have O minus, so we have a negative charge. So we follow this flowchart, and then we can see what the protonation state will be on the amino acid or the polypeptide that we're looking at. And so let's take a look at glutamic acid at neutral pH, right? So that means that pH equals 7. We compare the values. So 7 compares to 4.07. It's larger than, so the pH is larger than the pKa, which means that the deprotonated form is favored. So the correct way to draw this molecule would be to have a minus charge on that oxygen there. And this is the oxygen on the R group. So this is how we know that this is a polar charged amino acid, right? This right here is the R group because this is the alpha carbon. This is the R group. If we compare the second group here, we see that 7 is going to be less than 9.4, which means that this is going to be in the protonated state. So this should be NH3, and it should have a positive charge associated with it. The C terminal has a pH of 2.1. 7 is greater than 2.1, so this should be, again, negatively charged there. So this would be the correct protonation state for glutamic acid at neutral pH. So what we do, again, is we compare the pKa and the pH values, and then we determine if it's going to be protonated or deprotonated. Then we look at what the protonated versus deprotonated states mean for the nitrogen groups, the amine groups, and the oxy or the oxygen-based groups, most likely the carboxylic acid groups. And that tells us how we should leave the oxygen or the nitrogen there. What should the charge be, right? And when we do this, we see that we get a number of charges that are on our amino acid. That's how we adjust the amino acid charges based off of the protonation states. And so here's some practice that I encourage you guys to go over and do on your own. Just for the sake of time, we're not going to go through every example here, but use the exact same methodology here, right? So this first one has a pH of 7.3 is what the solution is in. The second one is a pH 3.2, so it's an acidic solution, and then the last one is going to be an alkaline solution, a pH of 12. And so we can also look at the isoelectric point of amino acids. So this is going to be mostly just for neutral, meaning polar uncharged, and polar uncharged and nonpolar amino acids, because once you add more pKa values in there, it gets a little confusing. And this is the pH at which the amino acid exists largely as a Zwitter ion. And so what a Zwitter ion is basically when we have charges on the molecule. So this isn't just for amino acids, but a net charge of 0, right? So when you add up all the charges, it goes to 0. And so you can calculate the isoelectric point just by adding the two pKa's on the amino acid, and then you divide it by 2. And this gives you the isoelectric point of the amino acid, the polypeptide, of whatever. Whenever you have the Zwitter ion form, that means that when the amino acid or peptide exists in the Zwitter ion form, then you have you're at the isoelectric point. And so you do the exact same thing we did here by adjusting the protonation states of the amino acids. And so going from amino acids, so now we talk about amino acids, we can then go to polypeptides, which are then polymers of amino acids. And they're held together by what we said covalent bonds, right? So these are covalent bonds. And they form peptide bonds, which is going to be a bond between the N and C termini of amino acids. And so this is usually requires a lot of energy for this to happen. So in the synthesis of proteins, we usually use ATP, which is the energy currency for the cell to synthesize these, or especially in the ribosomal unit in the cell. And so basically what we have is if we have two simple amino acids, let's just say we have two glycines, right? We have H3N alpha carbon C terminal, and this is what our glycine looks like, right? Because the R group again was the R group was the hydrogen here, and then we take another glycine and we duplicate this. This will then come together to form a polypeptide because the C terminal and the N terminal will then bond to each other. So we'll have an amide forming is what the functional group is going to be. So then we'll have H3N plus. Generally, when these reactions are happening, we're happening at uncharged species, but just for the sake of keeping things consistent with neutral with neutral pH, I'm just writing the charges here. And so these come together to then form the polypeptide. And so you see in the polypeptide, we only have one N and C terminus because the other N and C terminus have come together to form a peptide bond. All right, so that's the peptide bond right there. And the functional group that's present is the amide functional group, which is what I just highlighted right there. That is the amide functional group. And the amide functional group is the main functional group for the protein. And this repeating unit on a polypeptide occurs all the way through the primary structure of the polypeptide. And what I mean by that is if we looked at each point, we have the amine, then we have the alpha carbon, then we have the carbonyl carbon, followed by another amine, followed by alpha carbon, followed by carbonyl carbon, right? So this is the same sequence throughout the entire protein backbone. We have the amine, alpha carbon, carbonyl, amine, alpha carbon, carbonyl, right? So this sequence can go on forever. And anywhere between the carbonyl and the amines, this will be the peptide bond, right? So this can go on indefinitely. So if you're ever looking at a polypeptide and you need to identify the different R groups, you can just identify all the alpha carbons within the polypeptide, and that'll tell you where all the location of all the R groups are. The N nitrogen parts, or the amine side of these amide bonds and peptide bonds, we notice that they also have hydrogens that are bonded to them. And so this hydrogen can actually participate in hydrogen bonding, and the carbonyl can act as a hydrogen bond acceptor. So this is how long parts of the peptide can interact with each other and form secondary structure, right? So this would be an example of secondary structure when you get this hydrogen bonding across far parts of the chain because it's actually folding and moving in three-dimensional space. And another thing about the peptide bond is because of resonance, so due to resonance, it has the peptide bond, that is, has double bond character. And so because the peptide has double bond character, it means that everything along the backbone is coplanar. So if I highlight this right here, this all exists within the same plane, and it's only the stereocenters, which are at the alpha carbon, that you would get the stereochemistry. Everything else is coplanar. Here we just have two glycines, so there is no stereocenters here because of the glycines, but this, everything along the backbone is going to be coplanar because of resonance. When we destroy the structure of proteins, this is called denaturation of the protein. And we're not going to talk much about enzyme functionality and whatnot here, but remember we talked structure and function are extremely important in proteins. If you denature a protein, this means that you lose its function. And so what causes proteins to lose their function and lose their structure? Well, we can have chemical reactions, and this would be hydrolysis, which we've talked about before with other functional groups. Here we'd have hydrolysis of an amide, right? You destroy the primary structure, you destroy the structure of the protein. We can have a change in pH. And so we can think about that because if we change the pH of the protein, we change the protonation states. Let's say we have two uncharged groups next to each other, we change the pH, now they're both charged, and let's say they're the same charge, they will repel each other. So they will then move away from each other. So that can destroy the structure, right? So this changes protonation states of the amino acids within the protein. A change in temperature. This can melt structure. Then we can have other more complicated processes like irradiation of light and then heavy metals that can form coordination complexes with the proteins or bind to enzymes and stuff irreversibly. And sometimes heavy metals are extremely acidic too, which then results in a change of pH. And so to summarize this, anything that interferes with intermolecular forces. Right, so anything that interferes with intermolecular forces can denature the protein structure. And so proteins have evolved to have a certain structure depending on the environment they exist in. So for example, trypsin is a protein in the stomach, and the stomach has acidic conditions. And so the protein has actually evolved to have protonation states that align with that pH. If you raise the pH of trypsin, it will stop to function as well. So I know a lot of people are into drinking a lot of alkaline water now, but there have been some studies now that have shown that if you drink a lot of alkaline water, you change the pH environment of your stomach, and the trypsin protein, which is responsible for digesting proteins in your body as well, stops to function as well because you're changing a pH in the solution. So that's where we'll leave it for proteins and amino acids. We didn't get to talk about enzymes as much, but you guys will learn about that when you continue on into biochemistry. Next, we'll talk about lipids and fats and oils.